Okay, so my name is Mattia Prosperi, and as I said, I'm an associate professor at the Department of Epidemiology here at University of Florida. First of all, I want to thank the organizers for giving me the opportunity to come and speak today, and also to explain the work that I'm carrying on here at the University of Florida that I carried before at the University of Manchester, UK, where I came from. Some formal declarations. I don't have any financial conflict of interest to declare, and then I can get started. Yes. All right. Does it work? Let's start. So the title of my talk is Multi-Domain Inference in Healthcare, and how we can build a big data community in healthcare and in health sciences. What this talk is not about is analysis of the most common high throughput and big data that we are dealing with in health and biological science, which is next-generation sequencing data. I will cover this topic, but not in detail. I assume that there is a whole processing and research pipeline that give us processed data from next-generation sequencing. If you're interested, University of Florida has a lot of course curriculums in next-generation sequencing data analysis. I myself organize a summer course every year. It's called the Viral Evolution and Molecular Epidemiology Workshop, so you can contact me if you want, and we can look into details of next-generation sequencing analytics. Okay, so in general, what about big data inference in healthcare? The first thing is that electronic medical records together with large-scale laboratory data, for example, genome-wide sequencing, they dramatically impacted all healthcare sciences in the last decade. The key word here is integrated databases, because first we looked into harmonizing and storing in electronic format all the healthcare information, but then we needed to integrate these. And the problem was that different data sources, clinical trials, general practitioner data, hospital laboratories, must be integrated. So you need this standardized system for electronic health records, which are not yet a reality, because the integration itself is partial and difficult. But the technology is now keeping up the pace. And most of commercial and open source AHR, electronic health record system, are somehow integrable, because standards, ontologies, and mapping between different software have been made. Now, a very important thing is that there is a transition from syntactic to semantic integration. The syntax is how you encode information. For instance, you use ICD-10 ontology. In UK, we use the SNOMED for clinical data. But the metadata is important. The settings where the data has been collected, for instance, or the design of a clinical trial. And these brings us to the second important keyword of big data in healthcare, which is study design. We have to think at multi-scale modeling. We have to think about multi-scale and layered modeling, because we look now at realities that goes from the microscopic environment, from the cell to the pathogen, to the host, to the system, host-to-system, pathogen-host interactions, to a population. And when we look at integrated big data, usually it's observational data coming from, say, clinical trials and other types of study designs. So we will have sampling biases when we integrate the data. And instead, inside the data, we will see that there is high dimensionality, sparseness of the data, heterogeneity, missing values which might not be missing at random. So the study design on big data becomes a new type of science where we have to integrate the different settings too. Suppose that we are able to harmonize and homogenize this step, then we go into the statistical inference. And obviously with big data you have thousands of factors that need to be screened, either with large or small population sizes. If we do a GWAS studies, maybe we have a few hundred of subjects and many thousands of factors, variables to analyze. On the other side, we want to suppose that we want to analyze population-wide in a regional setting. Say we analyze all the people that were diagnosed with chronic kidney disease in either UK or in a US state. We have thousands and thousands of patients and a set, maybe reduced or maybe large, of clinical codes. So in order to process the data and also to fit statistical models, from linear regression to very complex modeling techniques like neural networks, random forests, often we need high-performance computational infrastructure and expertise. We also need computational efficient algorithms. We cannot parallelize everything, or sometimes even with parallelization the computational complexity is too much. When we analyze next-generation sequencing data, there are well-tuned algorithms geared towards these type of data that must pre-process the data and then give to the, for instance, to the epidemiologist clean data to make use of. So at this point, supposing that we have this efficient computational infrastructure, we move into the model and variable selection and the prediction performance and the validation of our model for predicting phenotypes. And at this point, when we have the best candidate model, we also want model interpretability. We don't want usually a black box. I mean, we might eventually end up with a black box, but we want to interpret how we predict a phenotype. And what's the meaning of all of these? Why are we doing this? We're doing this for precision medicine. In the past 2 years, precision medicine substituted the term personalized medicine, but it's basically the same thing. We aim at a so-called health avatar. We want to substitute, a virtual substitute of a person that describes this person's background and helps in optimizing diagnostic processes. This is also not only at the personal level, it's also at the population level. We want to improve the population health. And it's also at a higher level, which is the patient-provider interactions. By elevating this health avatar at a working level, where the patients can also look at these virtual representative of themselves, we can facilitate the patient-provider interactions. If the patient understands how the diagnostic processes work and can be automated, then we can improve also the trust. Now, I'm the first today speaking about big data, and the people that are investing and studying big data are also finding very evocative terms and definitions and so on that have been evolving also through time. So big data was initially defined with 3 words, with 3 V's, which were volume, variety, and velocity. Volume means data size, variety means different forms of data sources. As I said, it could be genetics, genomics, other omics, clinical data, surveillance data, and other types. Velocity is the speed of change, so how these data types change and how the metadata change. And veracity, which is a strange term, but means uncertainty of data. But then new problems came, so the V's became 4. They added, the V's came 5, and they added new V terms like value, viability, viscosity, virality, and different interpretations of these. So with big data, the scenario, the context is also evolving. And we can see these, I mean, this has been called as a fashion runaway. What is a typical or atypical research scenario? So we want to predict a phenotype, any type. This is our endpoint. For instance, we want to diagnose a pathologic condition. Phenotypes can be both objective and subjective. Objective could be cancer therapy failure, which can be directly measured. Cancerous tissues, or the fact that after a treatment against the virus load rise up. So it's objective, it's measurable. But there are also subjective phenotypes, and today I will give an example of these subjective phenotypes. For instance, differences in doctors' diagnosis. One prime example is asthma. On maybe antibody testing for infection, which can have a sensitivity and specificity variable. So once you define the endpoint, and you must be careful about the objectivity and subjectivity of these endpoints, then you define your study, you design your study, you look into all possible biases and so on, integration of different sources, and you come up with your data and variables. And you can have different sources, for instance, demographics, variables like date of birth, gender, risk factor, ethnicity, things that are collected in a routine clinical visit. Laboratory markers that might not be taken at a clinical visit, might have been taken in different sites from doctors' referrals and so on. So here you start having kind of missing data, the fact that the doctor requests for a test and another doctor does not. Bilirubin, glucose, T-cell counts, viral loads, HLA typing, anything. Then you have environmental variables. Variables and factors, information that has been collected outside of the healthcare facilities. This could be collected by survey. This could be collected by sampling. In UK, we had an asthma study where nurses were going into houses sampling for dust mite and other allergens, toxins, or pollutants. So collecting environmental data might be subject to systematic error. Then there is the data from next-generation sequencing, from microarrays. That could be either raw data or could be already processed data. For instance, out of a GWAS experiment, we get a set of candidate SNPs, single nucleotide polymorphisms, that have been previously associated with our endpoint or are kind of in a region that could be associated to the endpoint. And then new types that are coming up, methylation data, microbiome, bacteriome, gene expression, and all the new data types that are coming out with the evolution of next-generation sequencing technologies. So all this data must be processed subject to a very specific and clean study design for the final use. And the data must be cleaned, imputed, normalized, and transformed and encoded into something that is integrable. We cannot really compare apples with oranges. Here we have to define a meta model that enable us to put together apple and oranges. There is also a new generation real-time data as it comes from online social media. For instance, lifestyle habits, diary habits, and this is very difficult to encode. Usually it's text mining. There is a whole field, a whole new field in epidemiology that is called infodemiology, information epidemiology, where big data from social media is mined to derive variables and informations that are useful to do epidemiology. For instance, rates of influenza or of alcohol abuse by mining, for instance, Twitter. And I want to show you an example, probably this is familiar to you. It's the parable of the Google flu algorithm. So Google in 2008, at the end of 2008, started an algorithm to predict influenza rates in the US with a very simple and efficient idea. They were counting how many people were querying Google for flu. And they had a very simple algorithm that in the first years was kind of reflecting the state of the art, which is the CDC estimates, which are controlled and pure and very reliable, although they are made on a smaller sample. But then there was something in 2013 for which the Google flu trend algorithm started to double the estimation of the incidence. There was actually a 6.0 point difference between the CDC estimates and the Google. And this was because there were unreasonable changes to the algorithm that were actually made by computer scientists, not by epidemiologists. And there was also an amplification effect for which also the algorithm was known. There were some factors on people's habits that were not accounted for. For instance, the amplification effect of thinking that someone can have flu or not. Was instead the CDC estimates were consistent. And actually in 2013, this was not the peak of flu incidence as compared to the 2009. So this big mistake was called the big data hubris, and there were a couple of papers, well, commentaries in Science that analyzed the problem. But I want to cite a couple of scientists' phrases that are really important. The first one is from David Laser from Northwestern University, and he said that the big data hubris is the often implicit assumption that big data are a substitute for, rather than a supplement to, traditional data collection analysis. So there are traps in big data, and these traps can be really, really relevant. When you do statistics on big data, your confidence interval can be very small. You can give very precise estimates, and these estimates can be completely off the reality. So how can we try to handle these hindrances? We can try to break down the problems, and we can still apply classical modeling techniques, subject to the fact that we don't have to fall into these big data traps. Things that I usually explain when I teach the modeling to the students is this three-way breakdown. The first one is called design and get. So you design a precise study which is subject to the information you have available. And it must be carefully designed, looking at the data integration and extraction procedures, possible biases, confounding factors, and missing value analysis. Which is, it is difficult when you have single source data. It's much more difficult when you have multi-source data, when you integrate databases. Then you start the inference phase. You have to choose a model or a set of models. You have to test this model on your data and on external data. You have to check if you can find a compact model that can be interpretable, versus the fact that you might have a more complex model that has higher performance but is less understandable. And then you want to compare against the state of the art. So what's out there? The third phase, if you're successful in the second one, is to develop and deploy. You want to create use cases, you want to implement software, you want to assure quality of the software and of the modeling, and then you think about commercialization strategies. And the way in which you implement the modeling phase is, can be simple or more complex. And here I have a picture that tries to show how you break down the select and validate steps. So if you want to follow me, the first block is your data. It's been derived from the study design, the black blocks, and you have a number of observations and variables. And suppose that you can abstract these like a data matrix. You have to look here at the number of observation independence of the number of variables. You might have a number of variables that is even higher than the number of observations. So you have to specifically address these problems. At this point, we assume that the data has been homogenized. For instance, we had a set of SNPs that has been already characterized with a statistical modeling with respect to the outcome. So they have been, for instance, testing an additive model versus another type of model of association. And at this point, we have been given data which is already a good candidate, which has been already filtered. And then we have encoded the data with other heterogeneous data. So we have made sure, for instance, that there is a reasonable way to assume the missing values at random. If not, we have to encode them as not at random. If we have different domains, type domains, for instance, real numbers, categories, and ordinal variables, we have to carefully encode each one and take care of them. If we apply principal component analysis on data that has been that is half binary, half numeric, or it's numeric, but distribution and the joint distribution does not meet our requirement, the analysis is going to give us results that are not usable. So after we have a good study design, data extraction, then we start looking first at the features. If we have more variables than observations, we have to select the features, and we can use filter methods that are before the models or embedded methods that are within the models. And then we start fitting statistical models. We can start from a single one-rule hypothesis, the simple hypothesis, one variable that is associated to the outcome, univariate analysis. And then we go into more complex models. We can fit a generalized leader model, but if we have 10,000 variables, we have to use a shrinkage method. We can try Bayesian modeling, naive Bayes, which is kind of an equivalent Bayesian version of logistic regression. We can look into more complex Bayesian networks that allow a nonlinear effect, and we can look into causal pathways. We can use decision trees, which are nonlinear stratified models. We can do a lot of decision tree mixing, and this is called decision tree ensemble, and there are some implementations like random forest or bucket trees. And then we go further into more nonlinear models like AdaBoost or neural networks. All these models must be optimized, so we have a coefficient estimate, topology optimization for the networks, split criterion for trees, and so on. There can be further variable selection after or within the model. And then after this model inference and building, we have the brown part, which is boxing everything, which is the validation. We want to see how the model behaves with unseen data. And then also we can analyze the model performance with different measures. One can be interested in the percentage of correct cases. Another one can be interested in the sensitivity or the specificity of the model, or in both. So we can use different so-called goodness of fit functions. Now, fitting this model can be computationally intensive, expensive. So we need an appropriate high-performance computing infrastructure. High-performance computing services have a cost, and they need specialized expertise. Whilst it can be easy to run an analysis using SPSS or R, it can be more difficult to distribute this analysis on a computer cluster. Even if you're familiar with R, you have to use specialized library for parallelization. Also, if your data is big, it might not fit to the memory. So you need data warehousing, integration capability, for instance, extraction, transform, load functions, SQL, XSD if you have XML databases, and so on. You need big data file handling. If your data is too big, it does not fit the memory, you have to use some technological solutions that permit the data analysis. And sometimes, for instance, you can apply linear regression into a big data file that can be read with a big data library, for instance, MapReduce HDF5. But sometimes you cannot apply another statistical model because the way in which the data is structured in the memory, for instance, is read by chunks, does not permit the application of that particular statistical model. So you have to tune and see all your range of, to see if all your statistical routines can be actually parallelized and applied on the data that has been read with big data reading functions. Some of them will be usable and some of them will be not. At this point, you can try to parallelize, so you can try to improve the scalability. And you can use different paradigms like distributed paradigm or using multi-threaded solution, large shared memory machines, and so on. So you need expertise for implementing these tools as a standalone or add-ons of tools that you have been developing so far. And also, you might want to have a group that enable web server capabilities if you want to, for instance, to have a platform that enable the remote usage and maybe with a graphical user interface that could be browser-based. So I want to show a survey that was results from a survey that we carried on more than a year ago now. So it's a little bit old, but it's important. We had a 4-month survey advertised in both professional and academic networks about statisticians playing with big data and big computers. It was only for statisticians. And not for statisticians that work with next-generation sequencing data, but for statisticians that work in public health or biostatistics. So we reached them using LinkedIn, Allstats, SAS, and Stata, R, SPSS forums, universities, and so on. We had not a big set of responders, only 100 of them, a little bit more than 100, 112. And 82% were from the European Union and North America. So the 70% of them were recently became interested in parallel and cloud statistical computing because they actually had to face big data. For instance, coming from population surveys or studies from integrated clinical records. However, 44% of them had absolutely no idea how to do that, parallel statistical computing. And few of them used multi-threaded routines or their computers. Some of them were citing the SAS distributed software, which is actually very transparent, but it's commercial, it's expensive. And a bit less than 30% of them successfully used high-performance computing infrastructure. So even if they had no idea how to do that, they actually were able to work with the help of somebody else. And almost 70% of them are dealing with gigabyte-sized data. So we also asked about the nature of their data. And 41% declared to have heterogeneous data. 50% of them coming from next-generation sequence, so it's everywhere. Almost 80% of them had problems with missing or problematic data, so the study design is actually a relevant, very relevant problem. And the majority of them would have liked to be trained or would like to be trained in parallel computing, but also would simply outsource their problems. Now there is a very interesting R that has powerful libraries for parallel computing. 60% of the people that were interviewed preferred open source software. Only open source was the people that were prone to use mixed, like commercial and open source. Only commercials were less than 40%. And the qualifications of the people that were interviewed were high. 54% had a PhD, 61% were senior postdocs or above, 81% male, and the age range was 30 to 40 years old for the 46%. At this point, I will show an example of a big data-driven prediction of asthma. Asthma is my example. And why did I choose asthma? Well, because I worked with that, but also because asthma is not really a disease. Asthma is a symptom. It's an umbrella of diseases. It's the most common chronic disease in developed countries. There is a relatively modest drug therapies, and actually, the way in which diagnosed is very subjective. To prove this, I can tell you that there are different guidelines to define asthma. European guidelines, UK guidelines, US guidelines. And usually the definition is, say, 2 or 3 symptoms out of 5. So if this threshold is reached, then asthma is diagnosed. One symptom is, did you have asthma before? So it's a recursive function. Other symptoms are like, did you wheeze in the past 12 months? or did you wheeze in the past 6 months? Did you wheeze outside of cold? So the person that is asked this question must know that they wheezed outside of cold. And so it is a symptom. Asthma is wheezing, is having problems with the respiratory tracts, and so on. In fact, it's like flu. Like flu is not a disease, it's a symptom of something else, of different diseases. So there is this increasing recognition that asthma is a heterogeneous disease. There are similar clinical manifestations, so it's a phenotype asthma actually. But there are different underlying pathophysiological causes. These are called endotypes. And asthma has a wide support from the Ministry of Health and other federal funding agencies. There are several integrated population studies worldwide. In Australia, in Europe, in Northern America, Southern America. And so these integrated population studies, most of them contain information from very large domain spectra. Asthma has been tried to be analyzed by looking at genetics, environmental variables, demographic, laboratory data, everything. So if we think how to integrate these different domains, then maybe this can help us in unveiling the mechanism of the asthma disease, not only predicting a single phenotype, but also in clustering the asthma into different endotypes. So the example I give you today is not about endotypes, but it's about predicting the phenotype, the subjective phenotype. So we aimed at identifying, using some machine learning techniques, to which extent this large-scale heterogeneous information can contribute and be combined together towards specific clinical manifestation of asthma. We used two subjective definitions of asthma. One was asthma, and the other one was a single symptom of asthma, which is wheezing, and then a related disease, which is eczema, which sometimes is used to diagnose asthma. So the study population was pretty neat. We had adults that were parents of children enrolled in a British cohort, the British Manchester Asthma and Allergy Study that was carried on in Manchester and the surrounding areas is the MAS cohort. It's an unselected population-based perspective-based cohort and currently counts more than 1,000 subjects. MAS is also a curated database where many fields are populated, many fields in the database. So it's an actual database. And these fields are populated by querying different electronic health record systems, from hospitals, GP practice, and there is an ontology. It's the SNOMED CT ontology, which is widely used in the UK, which is partly mapped to ICD-10. So for instance, if we wanted to integrate MAS with a US cohort that use ICD-10, we might have problems in the mapping of the clinical variables. For our study, we had pre-selection of about 1,000 attributes, so almost the same number as the number of subjects. They were non-genetic. These attributes were selected from more than 5,000 variables that are well curated in the MAS database. And these were selected by hand, picked up by hand, speaking to the clinical scientists. They actually had selected originally 223, and we extended the set to 1,000 of related variables. And these included questionnaire data, laboratory data, clinical data, and environmental exposure. So many different domains. And these were, all of them were carefully encoded. And then we had some genetic data. We had 215 single nucleotide polymorphisms that were previously screened in genes that had been associated with asthma. The GWAS study was a little bit old. It's a 450K SNP chip from a few years ago. But and the analysis were made by statistical geneticists before. So we were given these SNPs. And actually, I was able to trace back the metadata, so the papers that explain how these 215 candidate SNPs had been derived. But in the database, this metadata was missing. We actually worked to include these. The SNPs were there as variables. So they might have been derived wrongly. We don't know that. So we had to work to link the SNPs to the studies that were published, that were showing how the SNPs were selected. So we grouped these attributes into so-called domains. We had the demographic and environmental domain, so questionnaires, surveys made to the patients, the children, or environmental sampling, which might have this systematic bias. Then we had laboratory data, which was measurement of lung functions and hyperactivity through machineries, for instance, breath tests and so on. We had microarray data, which was allergen sensitization, either microarray or actually skin prick tests. It's been shown that they have a good correlation. Microarray data usually give about 100 different allergen IgE values. The skin prick test is usually 6 to 12 because you have to test on an arm. And then we have the genetic SNPs. The outcomes, as I said, were asthma, wheeze, and eczema. For the statistical modeling, we had this cascade selection. We started from the most important rule, and then we had logistic regression, which was shrinked because we had more than 1,000 variables. Then we had decision trees, random forest, and AdaBoost. So the complexity goes from very simple to very complicated. And we looked at the area under the receiver operating characteristic curve as characteristic as a goodness of fit function, which is highly interpretable and gives you at the same time a trade-off between sensitivity and specificity. We validate the model using Bootstrap, and all the analyses were coded in R, and we encoded these using parallel libraries. And we ran that on the cluster. So the first set of figures that I give you is how we selected the model. So we had to compare the statistical learning techniques, and we used all the variable domains all together. And we found out that a nonlinear model, which was the random forest, consistently dominated all the other methods. Although a linear method was not behaving too bad. So if this linear function was compact, so it's more interpretable because you have basically odds ratio of the variables that have been selected. If the performance is clinically okay, so if the doctor is okay with sensitivity and specificity, we might substitute a very complex nonlinear model with a linear one. And these estimates were done on Bootstrap, on the out-of-bag data. So this is data that has not been seen by the modeling. This is how the model should behave on unseen data. The higher the curve towards the right, the better the model. So the green one is the random forest, and the blue one is the linear regression. As you can see, they are very close. Actually, the difference in performance was statistically significant, but the p-value was 0.15, so it was borderline, very, very borderline. Now, we fixed the model, so either a random forest or a logistic regression. These results are from the random forest. And then we asked ourselves, what if we use single domains? So what if we try to predict asthma, wheeze, or eczema using only genetic data, only demographic and environmental data, only lung function measurements, only allergens, or all together? Well, first of all, when we fitted the single domain models and all the integrated one, we found out that even using everything, it was very difficult to predict eczema well. So probably we don't have enough information to predict eczema. But aside from these, look at the asthma picture, which is the most relevant one. If we use only SNPs, even if we have SNPs that had a very, very, very low p-value adjusted for Bonferroni 10 to the minus 7, 10 to the minus 9, when we actually were trying to predict the phenotype, the area under the ROC curve was just a little bit more than 0.5. It was 0.53, 54. So actually, almost like giving a random prediction. So they are not good to predict. There is a significant association, but there are other confounders that aren't taken into account that don't help us in predicting asthma. If you get the GYs from your blood, you can't predict if you are going to develop asthma. Oh, by the way, these were children between the age 5 and 8. The second domain is demographic and environmental, the second worst domain. So if you ask about familiar history, if you go into a house and you sample for toxins, dust mite, and other pollutants and toxins, you can predict better than doing a genetic test. It might be even cheaper. You just need to fill in a questionnaire. But still, you can do better. If you measure lung function, different breath tests, even some terrible tests like metacolin challenge, where you have a child breathing kind of an exhaustion pipe until they don't they don't feel bad, and then you measure when they feel bad. You can have a better performance, and actually you can start to have something which is clinically relevant. It might be used for diagnosis, the onset of asthma. Interestingly, when you do allergen sensitization, you get the best performance. But we also found out that those that had allergic asthma were different from the groups that had good prediction from the lung functions. They are highly correlated, but there was a subset that could not be explained by allergen sensitization. So that's another type of asthma. In fact, by putting together all these 1, 2, 3, 4 domains, so we used all these variables together, we were able to increase significantly the performance. So if you want to predict asthma better, you have to test the lung functions. You have to test the allergen sensitization. You have to do questionnaires. You have to sample the house. And you can do genetic testing if you want, although at the moment it's not really well suited. Indeed, we also found a new predictor in these 1,000 variables that had never been associated before with eczema, which was the whole body impedance. It's a measure of electric resistance of the skin. So that was an interesting finding. And I think that I'm ready to wrap up everything. So we can say that at the moment, big data inference in healthcare is real. I mean, this wasn't really big data. We had 1,000 patients, 1,000 variables. It's not really big data. It can fit actually in a single computer. However, running all these models took time, especially doing the cross-validation and the bootstrap, and fitting all the model parameters. So it is big data. The complexity of the algorithms was high. Also, we had 1,000 variables that were actually screened out of 5,000, and the SNPs were screened from 450,000 variables. Sorry, the SNPs were screened from 450,000 variables. So we came from big data. At this point, the take-home messages are: first of all, we have to be careful not to fall into these big data hubris mistakes. Study design, as we showed also by the survey, is extremely important, as well as data integration. We need to concretize this HHR integration. Well, the data will be larger and larger and more heterogeneous. We need to approach the metadata and the semantic integration in a meaningful way. We need to know how the data and where the data came from, how it was collected, how it was processed. And when we do phenotype prediction, we need to approach problems using the multi-scale and multi-domain modeling with a careful design. Running the models, we need to tune up and empower the existing statistical methods, not only to identify the prognostic factors or endotypes of phenomena. I mean, we can do clustering besides regression. And these models must be able to predict the outcomes well, with good performance. If automated diagnosis is our objective, we need good models, otherwise we use doctors. And when you do modeling, you have to identify companion approaches to investigate also nonlinear, more complex dynamics. It's not always, nothing is always sometimes solvable with a linear model. Sometimes the dynamics, there might be interactions, and this explodes the computational complexity. However, we are more keen to adopt models that are interpretable rather than black box machines. If we have to choose, we prefer the model that is more compact, easier to interpret. Actually, decision tree is just I mean, the doctor could follow some decision pathways easily, but sometimes we can't do that. And the models must be delivered with good population-wide prediction performance. So the generalizability is important. And in order to do that, we might need huge computer power, very smart optimization algorithm. Last two things. The first one is capability to implement software and deploy it in a usable way for accomplishing this translational objective. If you have a good model that has been implementing a very, very bad tool, it's not useful. And this is to implement, to reach this personalized precision medicine goal, which is the health avatar. And in order to do all of these, I think that interdisciplinarity is the key at the moment. Let me conclude with some thanks. I want to thank my old colleagues at University of Manchester that set up the mouse study, and then University of Florida Research Computing that is giving us the very high, big computational power, the Department of Epidemiology and all my colleagues, the Emerging Pathogens Institute, and I haven't written this here, but Cancer Genetic Research. So we have contact information, and the slides will be available to the public. We have some stats about the gators. I hope these are updated and how we are proceeding forwards to the future and where we stand. And thanks. Causality. Okay. So how causality is handled and managed or in big data? Well, it's like with small data. When you have longitudinal study design, it's easier to set up a causal model. When you have cross-sectional data, it's more difficult because the problem of reverse causality. Even if you use, if you approach the problem with Bayesian networks, and you have a very good model that tries to find the best topology, sometimes you have equivalent topologies for which one could have a sense in the causal way, and the other one could be equivalent in terms of performance, but could completely mess up the causal pathways. So we still have the same problems. We have more problems because sometimes the way in which you collect cross-sectional data or longitudinal data is more confounded by the fact that the data could come from different studies, or you could have underlying biases. So the problem is there, and it's even more problematic than with data that has been collected under unique study design.
What's the heritability for asthma? What are estimates of heritability for asthma?
In terms of SNPs? So there are several studies that associated genetic traits with asthma, and these have been confirmed in different cores, the same. So the SNPs that are used in this database actually have been confirmed in other cores, intercontinental cores. US has confirmed this, and the statistical significance is high. The populations are also well represented. I mean, we have thousands and thousands of especially children that have been tested. So there is a genetic component, definitely. Probably it's triggered. So it's something that is going to happen only if the appropriate environmental conditions are there. This is my interpretation. Yeah, well, this is interesting because with migrations of populations, actually asthma can be, the asthma incidence could be increased because suppose that there is a population that, so there is this genetic inheritability and the population adapts to an environment. For instance, I mean, we are exposed to allergens all the time. If we live in the countryside in Europe or in Italy, there will be some plants, some pollens that we are exposed, and the population, I don't know, across 1,000 years could adapt to this so that the asthma is not triggered because the genetic components are well adapted to the environment. Then you move a family in a completely different environment, which is exposed to a completely different set of allergens that for which there was no adaptation, and this get triggered. Anecdotal experience, my son, I moved to UK and then to US with my son, and when we went to UK, he got eczema and other respiratory problems, and I am sure it was the black mold under our carpet. Then we moved here, it disappeared, and he was born here. But I'm from Italy, my wife is from Korea, so I don't know what's his genetic background. I think it's very complex. We can guess, we can make we can have some ideas. I think that there is a level of adaptation. I don't know what's the extent of that.
Any other questions? This is the last question.
We did that. There are a couple of papers you can look at. So we tried to do angiotype discovery in asthma. We didn't use actually the phenotype, so the doctor's diagnosis. We looked at domains and we tried to cluster into 5 patterns within and across domains. And we found out that there are different specific groups that all end up with the diagnosis of asthma, but their variable patterns are different. That's why I told you some of them are mostly allergen driven. Others are not allergen driven. So there are people that are not sensitized to allergens that have been associated with high incidence of asthma, but still get it. And we have some signal which is explained by other variables, for instance, the lung functions on blood tests, eosinophils, and other indicators, but it can't be really explained. And this is actually the problematic population that does not respond to treatment. I mean, treatment on steroids. So if it's triggered by allergens, you get Zyrtec or other drugs you can get better, or there are sprays, the inhalers, and so on. But some of the population does not respond because it's probably a different asthma.
Thank you. Wonderful. We're going to get started. It's a great pleasure to introduce you to John Hickey. He's now at the Roslin Institute at the University of Edinburgh as a career track fellow. John has been working with a number of different things, plants, animals, humans, so I think he really brings an interesting perspective here within this meeting. He's been a little bit all over the world, working at CIMMYT in Mexico, but he got his PhD and his undergrad in Dublin. Right? And then in addition to doing his postdoctoral work at CIMMYT, he also was in Australia at the University of New England. So, thank you, John.
Thank you, Matthias, and thank you for the opportunity to come here to this meeting. I've watched the meetings on the web for the last few years, and I really think it's a great initiative, and I think we, as a community, we need to have more of these kind of web-based delivery meetings, and also the idea of incorporating animals, plants, and human genetics together into a single point is actually very good. So I think it's really good. So I'm going to talk a little bit about a concept, let's say, or just to give it a name so that we can distinguish it from something else for the purpose of discussion, what we're calling Genomic Selection 2.0, which is how we will derive breeding benefit, in animal breeding benefit, from millions of animals with sequence. And essentially we're thinking in terms of, with these millions of animals with sequence, we will reach an asymptote of accuracy So we're no longer interested in increasing accuracy. We're interested in deriving genetic gain from other components of the breeder's equation. So I'm going to give an overview this thing works? Yes. I'm going to give an overview of animal breeding in terms of the basics and what has been achieved, talk a little bit about genomic selection, and then talk about this concept of genomic selection 2.0, and focus on three components of that. Firstly, how we will generate millions of animals with sequence information at low cost, how we will utilize this data to drive genome editing for complex traits, and how we will use the manipulation of recombination rate to release more of our standing genetic variation in our population to enable us more efficiently turn our genetic variation into genetic gain. So I think a lot of people will already know this, but just to start at some point, animal breeders are interested in response to selection, where response to selection is the difference in the mean between two subsequent generations. So we have this distribution of breeding values, we select some portion of individuals, and then by doing that we shift the mean in the progeny. And across many generations, for example, we can double milk yield or some other trait. The current gold standard way to do this is genomic selection to estimate the breeding values of these individuals. So if we think about how we use, or how we do drive response to selection, we have the classical breeder's equation where we have four components: accuracy, selection intensity, diversity, and time. And if we think, I like to picture diversity in terms of haplotypes, where we give each haplotype in the base population a unique color, and then across time we mix up these haplotypes such that each individual at some point has some mixture of colors. If we took some set of individuals down here as a distinct generation, we could encapsulate the genetic variation in that into some metric, for example, genic variance. And then we could say, well, we have 100% of our genic variance at this time point, and then with some degree of efficiency, we turn this variation over time into response to selection. And the efficiency with which you turn this genetic variation is quite important, into gain is quite important, because if you utilize it too quickly, maybe you rapidly make genetic progress, but you reach an asymptote quite quickly. And if you're more prudent about how you turn this variation into gain, you can ultimately reach a higher point at some time in the future, but maybe it takes you longer to get there. So animal breeding, just like plant breeding, has been quite successful, and one of the classic examples of the benefit of animal breeding has been the chicken from 1950s, which took, with 1 ton of feed, it could produce 330 kilograms of meat. The chicken from nowadays can produce 590 kilograms of meat for the same amount of feed. So this is a massive improvement. In addition to the quantitative genetics that underlie the genetic improvement in our populations, we have very good dissemination systems that globally disseminate this genetic gain. And I think it's just important to point this out, because I think it's quite an achievement of the breeding industry. So typically they're organized into, depending on the species, but in pigs and poultry, for example, the supply chain of genetics is organized into 3 layers. We have a nucleus layer, which has hundreds to thousands of parents per year, a multiplication layer, which takes the genetics from these individuals and multiplies it simply, and that would have hundreds of thousands of individuals, and then a production layer where farmers are growing the progeny of this multiplication layer, and we have millions or hundreds of millions of pigs and chickens, for example, grown globally by farmers. So if we take the global footprint of one individual from this nucleus layer, so after a 3 to 4 year period where we do this multiplication, one individual from this nucleus impacts the genetics in 3 million chickens, slaughtered chickens. So it is a huge impact. So any improvement we make here is massively multiplied into increased productivity, for example. So the gold standard way to drive this increase in response to selection is genomic selection, and I think David has explained it quite well, but I'll just go back over it because I want to make some distinctions to prepare for later in the talk. So like everybody knows, we have a training set, which is some set of individuals that are genotyped and phenotyped. We train some prediction equation, which is a black box, which has no, let's say, biological interpretation, and we can use this black box prediction equation with a set of individuals that are just genotyped to have accurate breeding values and then select the best individuals. It is really important, as David said, how you design this training set. So it can be quite small if it is very closely related to the individuals you want to predict, or it can be very large and therefore can be used to span a greater, let's call it, relationship horizon. So the original model as proposed was built upon the understanding that people had of quantitative traits at that time. And if you look at the paper, I think they assumed that a typical quantitative trait was controlled by 100 QTN, and the effects of those QTN were coming from a gamma distribution, such that about 10 of the QTN explained, I think it was 90% of the genetic variation. And if this was correct, then you would just saturate the genome with 50,000 markers, for example, and have some variable selection model that would pick out some small subset of the markers, which would capture the effects of these QTN, these essentially the 10 QTN, and with that then you could train from very distantly related individuals and a relatively small set of individuals and predict these individuals. But what happened in practice and what we have learned in the last 15 years, let's say, is that quantitative traits are much more complex than this. I think a lot of people would accept that there are thousands or tens of thousands of favorable alleles that control quantitative traits, and each of them have very small effects, such that we would need huge amounts of data to be able to estimate their effects individually. And despite not having the required huge amounts of data, we're still able to get very good predictions with the type of genomic selection that has been used in practice, because essentially what is happening is we are constructing training sets where we have subsets of very closely related individuals to the individuals we want to predict. These individuals share long haplotypes, so let's say 10, 20 centimorgan chunks of DNA. You just need to be able to estimate the effects of these big DNA segments, and then with them predict into the candidates you want to predict. And as long as those segments don't get broken up by recombination, the accuracy persists. And in practice, we are getting it in some of the major national animal breeding programs, such as, for example, the USDA dairy evaluation. I think their current accuracy for total merit index is 0.86, if I recall correctly. So extremely high accuracies of genomic prediction. So in some senses, you might say we've reached an asymptote of genomic prediction, and then we should just stop and move on with something else. But that is true, I think. But we could also say our 0.86 accuracy is coming from this black box that has very little precision within it. And instead of using a kind of a prediction based on these large segments, if we had millions of animals with whole genome sequence information, maybe we could shine some amount of light into this black box where we could have our predictions being driven largely still by this black box, but some of it by LD between QTN and causal variance, and in some cases by the QTN, the causal variance themselves. So again, just to remind ourselves why genomic selection has been attractive to animal breeders. So the breeder equation has got 4 components in it, and genomic selection directly addresses 3 of these components. So if we take accuracy, the accuracy of a breeding value for an individual without any phenotypic data is around about 0.5. With markers, it's now around about 0.8. Of course, this is still lower than the 0.9 we would have obtained if we had done a progeny test on that individual. But it takes 6 years to do a progeny test, so we can get 0.8 in 1 year via genomic selection, or 0.9 in 6 years via progeny testing if this is a dairy cattle situation. So you're saving time, and in some sense, you're addressing accuracy. You're also addressing selection intensity, because to get this progeny test, maybe it costs $40,000, and with genomic selection you can get a 0.8 accuracy for something quite a bit less than $100, depending on your genotyping budget or your costs. Additionally, the $40,000 phenotype maybe only reflects some of the traits that we're interested in. So, for example, maybe milk yield for early heifer performance, for example. So it does not encompass the whole lifetime economic value of the performance of a cow. So genomic selection can give us accuracy for a broader range of traits that are hard to measure at an earlier stage. So genomic selection has been adopted now by all the major livestock breeding programs globally, I believe, and it has been a major success. So essentially accurate breeding values are possible. Shorter generation intervals have been achieved in the species where that was the attraction. How genomic selection works in terms of how you need to design a training set, what marker density you need, etc., are well understood. Good systems are in place to record the data, to do the genotyping, to do the imputation, etc., to drive the predictions. And I think most importantly for the future, breeding programs can now generate lots of data. So large breeding programs will be genotyping 100,000 animals a year. Some portion of those will be phenotyped. So we could be growing training sets quite rapidly and growing numbers of animals that are genotyped even more rapidly. So in that mind, then we sort of say, well, we could continue with our 0.86 black box accuracy based on capturing linkage segments, and we could be very happy with that. Or we could say, let's go and sequence everybody and see if we can drill down more precisely into the genomic predictions and try and get at these QTN, and then with that information try and drive extra genetic gain through some other route that is not accuracy. So David already talked a bit about the impact of relatives, so I just want to talk very briefly here because it sets up the next slide. So everybody knows relationships affect the accuracy of response to selection, and we have a simple way of parameterizing that. So we had a training set of something like 2,000 individuals, and we had the set of selection candidates, something like 509 selection candidates, I think. And when we looked at the accuracy of these guys, we saw that they spanned from very low of order 0.15 to very high of order 0.8, even though we had the same training set. And then we just looked in this training set and saw that the individuals with the high accuracies, or in the prediction candidates with the high accuracies, had the equivalent of 10 full SIBs in the training set. These guys over here would have the equivalent of 10 half SIBs in the training set. These guys would have the equivalent of 10 cousins in the training set. And what we really want to do is take this red line and pivot it on this point and shift the whole thing up such that we have highly accurate and highly persistent predictions all the way across this, let's call it, relationship frontier. Perhaps it's not entirely possible to do this, but this is, if you like, what we would ideally want to have. So just to distinguish that from classical genomic selection, we're calling this highly persistent accuracy genomic selection 2.0. Not for any other reason than just it helps me communicate the thoughts. And why this genomic selection 2.0 is, let's say, better than what we currently do. So this is if we take this as our typical animal breeding program with some nucleus up here where we're doing recurrent selection as quickly as possible based on genomic breeding values. And then we're pushing these genetics down through the multiplier layer into hundreds of millions of genetically distinct, or 100 million or so genetically distinct individuals that our producers are using, growing. So the problem with this approach is that the nucleus produces, let's say, 100,000 candidates per year, but nucleuses by design are limited in size, so let's say of order 10,000 individuals or 5,000 individuals, some small number of individuals. So the accuracy that you can get from this has a limited amount of refinement or precision. So it might be reasonably high, but it is not broken down into these QTN. And because of that, the predictions don't persist down here. And if we think about these animals, these are highly valuable individuals, so they're kept in high health, highly controlled environments, which are quite different to the production environments down here. So the phenotypes that are being recorded and selected upon are essentially different to the phenotypes that the farmers are using. In an ideal sense, we would collect phenotypes down here at the producer layer and use those phenotypes to drive the recurrent selection up here. But we can't do that currently because the accuracy of our genomic selection wouldn't persist across this frontier. So if we could get a really cheap genotyping platform, for example, $6 an individual, then we could start to genotype these producer animals where the phenotype is really what we are trying to improve, and use that phenotype to drive the responsive selection up here. So we would no longer need to phenotype up here. As well as increasing the relevance of our phenotype, we would also be able to identify, so in here we have, let's say, 100 million genetically distinct individuals down here, per year. Here we would have 100,000 selection candidates per year. So you have many more potential selection candidates down here, which means you could crank up selection intensity quite a bit if you could do your training down here. So there are other reasons why it would be beneficial. our hypothesis is obviously that sequence data has huge potential in animal breeding, but to realize this potential we need huge volumes of sequence data, because essentially you have however many SNPs there are in pigs. For example, I think the recent report I saw from humans where there are 80 million SNPs in humans. So if we're trying to estimate the effects of 80 million different segregating variants, we need to have lots and lots of data to be able to get enough recombinations to separate these things so that the statistical model can separate their effects. But the idea of getting 1 million animals with sequence information is actually within reach. We have a project in which we will generate 325,000 animals with sequence, and that project starts this year. And I think just this morning on Twitter I saw there were a human genome sequencing paper was published in Nature Methods with 1,000 genomes. And I think that's quite interesting, because if 1,000 genomes only gets into Nature Methods now, that means we're quite a long way down the road to generating huge amounts of sequence data. So with this huge data set, we want to do what we're calling industrial scale fine mapping, where some proportion of the genetic variance gets fine mapped directly to the causal variant. And the proportion actually we don't think needs to be that high. It doesn't need to be 50 or 100% of the variance. It could be 10, 15, 20% of the variance. And if we can fine map those, we can use them to drive genetic gain. So the benefits of such an approach would be we can do industrial scale fine mapping and biological discovery and utilization of our traits, let's say. We can capture and utilize the effects of de novo mutations, at least perhaps with some time lag. And this is interesting because current genomic selection kind of ignores de novo mutations, and we are selecting without using these de novo mutations, which ultimately is probably meaning we are approaching the exhaustion of genetic variance over time to some degree. We'll have more persistent predictions over many generations, so even though the accuracy will have asymptotal with both black boxes, this one will be more precise in some senses. And two things I'm going to talk about now will be how we could use this more precise information about our QTM to drive genome editing for complex traits, or to enable the manipulation of recombination, increase our response to selection. But before I talk about that, I'm going to talk briefly about how we will generate the data. So this is my very simplified view of sequencing. So we have some individual that is diploid, and we can do some X sequencing on this individual. So 1X will be 1 read, 2X 2 reads, 20 to 30X is typical in some applications. So if we take the sequencer and it would chop up the DNA, and if we focus on this segment here, we could get a 1X read which would read the blue piece or the red piece, but obviously never both. A 2X read could read the blue piece twice, the red piece twice, the blue or the red or the blue. And the more times you read this, the more X you do, the more opportunity you have of seeing both gametes. And in this case, that's important because you can see that this individual is heterozygous at this position. So if you were to follow that approach, then you could have this family in which we have a sire who produces these progeny, and if we look at the segregation or the Mendelian inheritance of these DNA segments, it could look like this. And we have the dam haplotypes of these individuals just grayed out because they're not relevant for the example. So if we do 10x sequencing of all of these individuals, we could end up with lots and lots of copies of the red and the blue haplotypes. And this is, in some senses, a waste of resources because it's treating each of these individuals as separate from the population, and it is sequencing them in complete isolation from the fact that they are related to other individuals. And an obvious example there is if we have one haplotype that we sequence in 30 individuals 30 times, then we end up sequencing this haplotype 900 times, and that's probably not good use of resources. So instead we say let's make the population the target of the sequencing resources rather than the individual. And if we do that, we would, instead of trying to get 30x sequence on each individual directly, we would sequence the haplotypes that are present in the population 30 times, but using the DNA from many individuals. And if we do that, we can use the sequence information on two sides of the, let's call it, imputation coin. Firstly, we can use the sequence information to derive consensus haplotypes. And then secondly, given we have these consensus haplotypes, we can reuse the sequence data to work out which combination of these consensus haplotypes each individual carries. And we think this system will actually lead to quite accurate imputation. And currently in livestock, we don't have tools that enable us to use this kind of data effectively. So we're left with using the hidden Markov models that have been designed for human data sets currently. And those hidden Markov models are very accurate in some senses, but they're not as accurate as algorithms that have been specifically designed for animal breeding applications, which take into account this linkage information, let's say, and pedigree information. And we know from classical imputation of SNPs that when you account for that information, you can get much more accurate imputation. But nonetheless, we used a hidden Markov model to compare, let's say, our low coverage sequencing strategy with a classical sequence, some small number of individuals at very high X and impute. So we have let's say some sequencing resources, 1,000x of sequence information that we can use at each marker in total in a population. So we have a choice then. We could say I'm going to do 25 sires at 40x and the progeny of these sires with 200 SNPs per chromosome and then impute. So that would be like a 6K chip if you were working with cattle. Or I can just sequence the progeny themselves at 1x, and then I can compare the imputation accuracy. And we get quite a big bump in imputation accuracy by taking the second approach. So there are two reasons why we get this bump in imputation accuracy. The first reason, obviously, is that this 1x whole genome sequence data, the information that you capture is much closer to the recombination locations, which enables you finally track the inheritance of haplotypes and therefore do good imputation. But secondly, the way in which sequencing machines sample DNA and read it is not uniform across the genome. So even though we're targeting a 40x average, for some markers in some individuals we get very low X, and that hits our imputation accuracy. And that's actually quantified here by comparing scenario 1 with scenario star. For scenario 1, as I said before, we have 40x sequencing on the sires and 200 SNPs in the progeny. Scenario star, we have infinite X on the sires and 200 SNPs in the progeny. So this difference in imputation accuracy is due to this unequal sampling, unequal amount of reads at each locus. And so in other words, classical livestock imputation algorithms work well with SNPs, but they do not directly translate. Their properties will not hold when we go to sequence data. So that was a very simple simulation. We also have something which we've actually produced these results last week, so they're quite recent. So what we have are we did a simulation where we have 4 generations of pedigree, and I think it was 500 females per generation, 50 sires per generation, and then producing 1,000 candidates in the 4th generation. So we could genotype the candidates with a 3K chip, or we could sequence them at some very low X and increase that X up to 10X. We could also have different percentages of the ancestral sires, so this 150 sires, we could have different percentages of those guys sequenced going from 0% up to 100%. So if we take this dot here, these are the selection candidates, let's say, being genotyped with a 3K chip, and all ancestral sires being sequenced at, I think it was 30X. So this could be the dot that you would get in a 1,000 bulls approach. So then you could say the gray line would be absolutely none of the ancestral sires being sequenced, and the blue line, all of the ancestral sires being sequenced. So in some senses, this is the LC-sec approach. So as we increase the coverage of the low coverage sequence information, I think when we get to 2x sequencing, we negate the benefit of doing any sequencing on the sires. Of course, this whole figure is dependent upon the number of candidates that get sequenced at this low level. So as you increase the number of candidates that are being sequenced at this low level, you can push this gray line up a little bit more. This was with 1,000 candidates. So then we had some other results where we said, in the absence of any ability to do imputation, how well would low coverage sequence data do in a diploid plant breed or animal breeding species for genomic prediction? So the plant breeders have been using 1x GBS data to do their predictions, but they were focused on these inbred lines, which meant that you didn't have this problem of failure to call heterozygote loci. And animal breeders have diploid outbred individuals, so we have this problem of heterozygotes. But we thought perhaps it doesn't matter, or at what X will it no longer matter? So we have some accuracy, we have some standard genomic selection training set and prediction set, and we end up with some accuracy of the breeding values as the correlation between the estimated breeding value and the true breeding value. We have some level of X that we can use on the training and prediction individuals, so going from extremely low X up to 20X, and then ultimately to SNP, and we just assume that SNP equals infinite X. Then we have different marker densities going from 3K to 300K. So if we just focus on the 300K data, we could get the full asymptote of accuracy of genomic prediction with 1X sequencing. So you don't need to be able to call these heterozygote loci to get really good predictions. As you reduce the marker density, you needed more X to achieve the asymptote. So with the 3K, you needed a 5X data. So that showed that GBS could work quite well, or at least we thought it could work quite well for a diploid species. But then there are other things that you can think about doing when you've got some controllable sequencing platform like a GBS, or I should also say a rapid sec perhaps. So we said if we just focus here on the green line, so we have 10,000 X that we can use at each marker position in our training set or our prediction set. And in this case, we're having a prediction set. So I can test, if I have 10,000 X, I can test 500 individuals genotyped at 20X average per locus, or I can do 50,000 individuals genotyped at 0.2X per locus. So if you like, this is really inaccurate genotype information per individual. This is highly accurate genotype information per individual, but we're playing with two components of the response to selection equation. We're trading off accuracy versus selection intensity. And our conclusions here were that the overwhelming thing to do was increase selection intensity at the expense of the accuracy of the breeding value. So now I'm going to talk a little bit about this page strategy. So genome editing is quite a hot topic, I think, in all of biology at the moment. And it's like all ideas that are good, it's a really simple idea. You can just go into the DNA, chop out some piece of it, and then replace it with some other piece. So in theory, you can add a nucleotide, you can delete a nucleotide, or you can replace a string of nucleotides. My colleagues at Roslin, so this is the second coming of Dolly the sheep. They have produced what they're calling PIG26, and PIG26 has a single base deletion in a certain DNA element, and this is actually a gene that's it's a candidate gene that the hypothesis is that it would confer resistance to African swine fever, which is a major risk disease in pig production. So they haven't proven yet that it does produce the phenotype, but it is important that they have been able to show that they can edit a genome of an animal and produce a live, viable animal. So then we thought, how would we take this type of technology and embed it into classical animal breeding, where we're interested in increasing the response to selection for complex traits. So the PIG26 was focusing on a trait that in theory was controlled by a single locus, let's say. But animal breeders are much more interested in traits that are controlled by tens of thousands of loci, for example. So in that case, maybe genome editing won't have a role. So we did a simulation to try and evaluate this, and it took us a long time to derive this equation. So we have a new equation for a new breeder's equation, let's call it. So we have the classical breeder's equation here, plus Page, which gives us genetic gain. And this is, just to communicate, this is important because we're going to do classical genomic selection, and then the individuals that get selected to become parents will be edited for some number of loci. So it is important to emphasize that in this strategy, which we call Page, it is about promoting alleles that already exist in the population. So it's not about bringing alleles from outside in. It's simply just increasing the frequency of favorable alleles and more rapidly than you can do with classical selection. The challenge, obviously, is we have quantitative traits which are controlled by tens of thousands of favorable alleles. You have millions of production animals, but the opportunities are we have this nucleus structure in which we have very small numbers of sires contributing to these millions of production animals. So in theory, you might only need to edit these guys. We expect to have at least huge genomic selection data sets which will be sequenced, which will enable us to fine map some causal variants, and we can use projects like FANG, which is the functional annotation of animal genomes, a little bit like an ENCODE for livestock project. We can use genome editing itself to, if you like, prove causality of our causal variants. So the objective was to quantify the genome editing resources we would require to have an impact on the rate of improvement for quantitative traits. How many alleles do we need to edit? How many per generation? How many animals do we need to edit per generation? What is the extra genetic gain that we would get? And are there any obvious downside of this approach? So we had a simulation, which is quite standard. We have 100,000 generations of random mating using a coalescent simulator, using standard parameters. Then we have what we're calling a recent historical breeding scheme, where we have 20 generations of breeding, where we're selecting using genomic selection with perfect accuracy in a recurrent selection scheme, where we have 1,000 candidates per year produced by 500 females and 25 males. So this is just a standard recurrent selection scheme. Then we reach some time point, which could be considered to be now, where we have a breeder, the controller of the breeding program has options for what they do in the future. They can continue with this selection scheme based on genomic selection, or they can supplement this selection scheme in which they would edit some portion of the sires that get selected per generation for some number of alleles, so 0 up to 100 alleles per sire. And it's important to remember that the trait we simulate is controlled by 10,000 QTN, and each of these QTN effects are coming from a normal distribution, so we don't have any mega QTN. So our comparison metrics are genetic gain, change in allele frequencies over favorable alleles, the number of distinct QTN that we will edit across our 20 generations, and this is important because if we had to edit 10,000 QTN, we probably wouldn't be able to find these QTN, and any impact there is on inbreeding. So this is the main result. We have the blue line, so we have generations here going from -20 up to generation 0, where we start our new strategies, let's say, and then 20 generations of future breeding. We have genetic gain up here when we set the base to generation 0, or generation -20. We have genetic gain up here where we set the base to current time, let's say. So the blue line is what we will get with classical genomic selection with perfect accuracy, so we're making some level of responsive selection. Then we can do QTN discovery at this point, find the QTN that segregate at this point, and then start to edit sires for the bigger QTN, or for the QTN that we choose in a certain way. So if we start to edit the 25 sires for 20 QTN, we double response to selection, and we double this response to selection at any point along this time horizon, let's say. And I think what's quite interesting is that if we look at the blue line, it's starting to asymptote. But if we look at the black line, we've started to remove this asymptote. So I think that's the second favorable feature of this strategy. So then we said, well, what underlies this extra response to selection from this promotion of alleles by genome editing? So when we do breeding across our 40 generations, we are trying to increase the frequency of favorable alleles. And the solid line here, the solid blue line, is when we do genomic selection with perfect accuracy, we shift the allele frequencies, the mean allele frequencies of all alleles, a very small amount. If we do the solid red line, it's what we do to all of the alleles when we do genome editing. We start to do this genome editing in conjunction with our genomic selection. But if we look at the dotted line, we see these are the shifts in allele frequencies for the 20 QTN from our 10,000 that have the biggest effect. So these are still small effect QTN, but they are bigger than most of the rest of the QTN, all of the rest of the QTN. And we see with classical genomic selection, we increase the frequencies of these bigger QTN to some degree, but at some point we reach an asymptote. And what we're seeing is that about half of the bigger QTN are being lost from our population due to drift and small effective population size and selection drag before we can use selection to fix them. We don't have this problem with the genome editing strategy, which is picking these big QTN and driving them to fixation very, very quickly. And obviously, once it drives the biggest 20 to fixation, then it takes the next 20 and the next 20 as we go forward. So this page strategy will be highly dependent on our ability to fine map causal variance. And if we were required to have to fine map thousands of QTN, it would probably be beyond the scope of what is realistic. So we wanted to quantify how many QTN were being edited per generation and then across our 20 generations of this process. So on average, we were editing around about 30 QTN per generation. Across the 20 generations, we were editing 314 distinct QTN. These QTN explained 36% of the genic variance in the base population. So if we extrapolate these numbers into what we are seeing empirically from the large human GWAS data sets, they're capturing 600. They have 600 GWAS hits, although a GWAS hit is not the same as a causal variant, I accept. And the proportion of variance that they're explaining is of order 30, 40, 50%. So while we perhaps don't have the capacity to fine map this exact number or this full number of causal variance in our planned data sets, we think we can get at least some of it. And we have a number of advantages in livestock. For example, so first of all, I think our family structures are more favorable to finding causal variance. And then we can use genome editing itself to prove the causality of candidate QTN. So genome editing is an expensive technology currently, so we thought some breeders may choose to only edit a subset of their sires. So if we're selecting 25 sires per generation, maybe they can't afford to edit all the sires, maybe they can only afford to edit the top 5 sires. So just to balance things, we said, let's assume we have 500 edits that we can do per generation. We can do 20 of those edits on all of the sires, or we can take the top 5 sires on the basis of their genomic breeding value and edit those 5 sires for 100 alleles, which is a little bit unrealistic to edit 100 alleles in a sire, but just to keep things balanced, we thought it was a good compromise. What we see is we get a massive increase in response to selection over our previous approach. So the blue line is the blue line we had previously for classical genomic selection. This green line is the line we had previously for response to selection when we were doing 20 edits per sire. So that we're getting this big increase even above this response by distributing our genome editing resources on a subset of sires. But this is also having a big negative impact because it's driving up the inbreeding quite a bit. So again, we have our blue line, which is inbreeding due to genomic selection. We have the red line, which is inbreeding due to genome editing when we edit all the sires equally. So there's almost no difference in inbreeding between those two strategies. But then when you focus the edits on the top 5 sires, you get this massive increase in inbreeding. And what's happening is we are taking the 5 sires and editing them, and then these sires' progeny become really much better than the progeny of the other sires, so that when we make the next round of selection, we're overrepresenting the grand progeny of these edited sires in the contributions that are being made to the next generation. So in effect, we're creating super grand sires that are damaging our population because they are driving up the inbreeding too quickly. So our conclusions is that PAGE is quite effective for increasing genetic gain, even if you can only do 20 edits per sire, and you can only need to edit 25 sires per generation, so relatively small numbers of edits and relatively small numbers of animals. If not managed properly, there is an increase in inbreeding. We obviously have a problem of target identification and target validation and off-target edits. This whole thing will fall down if epistasis is a problem. I've seen some empirical results which suggest epistasis might not be a huge issue. To make this work in practice, obviously we need huge data sets and good targets, and we need the costs of genome editing and multiplexing technologies of genome editing to be sorted. So I don't think it's currently possible to edit 20 QTN in a sire. It is possible, but you would need to use cloning and cell lines, and that will increase the generation interval. So this approach needs to be developed where you don't need to do that, where you can directly edit into an embryo. I think PAGE works because of its precision. So a weakness of current genomic selection or all genomic selection with, let's say, even if it has perfect accuracy, is that the alleles do not segregate independently, so you can't shift these alleles independently of each other, so you're having this constant collision between increasing some alleles and other ones are pulling them back through their linkage. But with PAGE, the alleles that are being edited behave as though they segregate independently, which is why you can have this very precise increase in response to selection. Can we find enough targets? I'm an optimist, and I think with 325,000 pigs with whole genome sequence information, we might be able to get close to this number of QTN and the portion of variance explained. But I do think classical animal breeding driven by genomic selection will obviously remain a cornerstone. So the final topic then is a little bit about the manipulation of recombination for increasing response to selection. And this is interesting for me because if we think about genomic selection, it addresses the accuracy of selection, it addresses the selection intensity, and it addresses the time, but it does nothing for diversity. And if we think about recombination, it is a rate-limiting factor in many aspects of genetics, and in terms of response to selection, it's a rate-limiting factor in terms of its ability to enable genetic diversity to be released and be selected upon. So if we start with this permutation of alleles, and if this is the idiotype, if we don't have recombination, we can never get the idiotype. So we need recombination to get the idiotype. Domesticated pigs have a higher rate of recombination than pigs in the wild. There is some evidence that plants that have been the progenitors of domesticated plant species have higher rates of recombination than plants that never became progenitors of domesticated species. And there is a huge body of literature in classical selection experiments that show, in Drosophila, for example, increased recombination is a correlated response when you're selecting on some other trait. So we thought, if we have perfect accuracy of genomic prediction, well, one way we could get at more response to selection is through the manipulation of recombination rate. So we have a simulation which is very similar to the one I described earlier. We have some ancestral population for some large number of years. Then we have some historical animal breeding where we're doing genomic selection with perfect accuracy. Then we have some future animal breeding where we're doing genomic selection with perfect accuracy. In the historical populations, we have a genome length, or we have 10 chromosomes, and each of the chromosomes has a chromosome length of 1 Morgan. In the historical population, we're selecting all the females and 2.4% of the males to become parents of the next generation. In the future generations, we are varying Morgan length, going from 0.1 Morgans per chromosome up to 20 Morgans per chromosome. And the red dot signifies that 1 Morgan stays the same, and in the figures that come next, you'll see the red line is, if you like, the baseline. Then we have, we can play with selection intensity, if you like. So instead of having 2.4% of the sires being selected, we can select 10% or 1.2%. Again, the trait was controlled by 10,000 QTN. So in this figure, I don't have the response to selection for the historical population like I did for the genome editing stuff. I just have the response to selection for 40 generations of future breeding. The red line is what we would get if we had 1 Morgan chromosomes, and then as we increase recombination up to 20 Morgans, we get quite a big bump in response to selection. And as we lower it, we get a reduction. So then if we look at the way in which we're utilizing our genetic variance, we have the red line where we're showing that we're losing variance, and ultimately we're going to reach some point where we lose it all, as we do with this really low rate of recombination. And with the increased response or increased recombination rate, we're seeing we're actually in the initial stages we're increasing variance. I think that's because we're releasing this trapped variance held up by Bulmer. But then we're also just utilizing the variance better, so we're losing it at a slower rate, if you like. So we have this figure then where we try to encapsulate the efficiency of turning variance into genetic gain, because ultimately that's what breeding programs try to do. So we have some portion, or we have some metric here, which is the used genetic variance in generation 40, and then we have here the response to selection in generation 40. Up along here we have Morgans of our chromosomes, and across here we have portion of sires selected. So if we see, as we increase recombination rate, we are always increasing response to selection, and we are always reducing the rate at which we're using our genetic variance. as we increase selection intensity, we sometimes are increasing response to selection, but we're also always increasing the amount of variation that we used. And then at some point we overcook it, and we utilize our variance too much, too quickly, such that we ultimately impact our ability to make long-term response to selection, which is why we see this drop-off. So I just want to particularly focus on these points here that are grayed out, where we have essentially what we consider it to be as the same rate of used genetic variation in generation 40, and this allows us to compare scenarios then for response to selection, and that's what's here. We have 4 different scenarios which have different, well, they use variation at the same rate, so by the time to get generation 40, they all have used the same amount. They have similar paths of loss of this variation, but they have quite different responses to selection. And in essence, what is happening is we are able to, with higher rates of recombination, we are able to sustain more intense selection, which is translating into more response to selection. So that is the final piece of topic I want to cover. I just want to thank some people. So Greg Gregorians is very much involved in a lot of this work. John Williams and Bruce Whitelaw are senior researchers in Roslin, and we are collaborating with Bruce in particular with this genome editing stuff. We've got support from Genus and Aviagen, and in case people are interested, we do have 2 positions which are opening up this month to work on. We have basically 300,000 chickens with sequence data, and we want to analyze it. I'd like to thank my funders. So the BBSSC is the British Biological Sciences Research Council. That's a major funder of research in the UK. Innovate UK is a major funder of agricultural research in the UK, and then we work with these companies. Thank you. So there is genetic variation for recombination rate. The Icelanders, lots of people have studied it, but the study I'm most familiar with is the Icelandic human decode stuff. So they have a heritability of 0.3, and they have found a QTN that increases genome length by 10%. So you can find QTN, right? So in these simulations, I haven't shown the results, but we tried to incorporate recombination rate as a trait in our selection index, and try and increase it and then translate it. So we were not able to get better response to selection for the target trait by doing that, because you were pulling off too much. You had to put so much selection intensity on recombination rate to get a big increase in recombination rate that it didn't show a benefit. So the only way we can see it working is if, well, there are 2 ways. One is we can genome edit the QTN that control recombination, or secondly, we can use genome editing type machinery to just edit, or to just break the genome and get it to recombine.
Compared to how transgenics are accepted in the community, how is it easier to convince people to use genome edited organisms?
So I'm not really qualified to answer that question. To be fair, I think that there are people that work in transgenics that are much more on top of these issues, so I just give you my opinion, right? So I sense that the transgenic stuff was not managed well when it was introduced. Not because the people did a bad job per se, but they didn't have experience. They were very excited about this technology, and they thought everybody else would be equally excited. So they just assumed, and then the world said no. I think with genome editing technology, they have had this lesson, so they're better able to communicate with people that this is a safe technology. So there is no genome edited animal in Europe or the US approved, I believe. There is this salmon that has been edited like 15 years ago that is, if they can get that through Congress, it could be a good step forward. Some of the bigger companies, animal breeding companies, are investing in genome editing. That's publicly known. And they wouldn't be doing it if they didn't believe that it would be passed. And there's one other component, but I forget what it was. Bottom line is, I think this technology is favorable.
So do you have an estimation of how much it costs, given that you're working with your colleagues there? And how much will it cost? And imagine that depending on that, you're going to have to edit up to a certain number of genes given the effect of those genes in the trait, because afterwards probably it's not going to be financially possible.
Yeah. So I don't have a number for what it costs to produce a pig with edits now, but it's a lot of money. But that kind of doesn't matter, because if you look at the genome editing news feed, there are companies in the human space and in the cell line space that are investing hundreds of millions of pounds in technology. So it's only a matter of time before an Illumina type company emerges with a multiplex, low-cost genome editing platform in the next 2-3 years, I suspect.
There's probably a balance between the allele frequency, the allele that you want to edit versus the cost, and you've got two options. The frequencies are around behind population. Simply through breeding, you might be able to bring it out to enough divisions that
Yep.
will be added.
Yep.
If the frequency is very low, then you really don't have a choice.
Yeah, we have, in our thing, we ranked the QTN for editing priority by effect size, but of course that's a silly thing to do. We would be much better to rank it by effect size times frequency. Yeah.
More questions for John? All right, thank you. Lunch is Patricio Munoz, of course. Most of you know Patricio. He was a graduate student here at UF in the plant life and cell biology program. During that time, he was working mostly with trees, but then as he started his postdoc, he started shifting more towards grasses and toward forage grasses. And this is where he's working out in an assistant professor position here at the Department of Agronomy here at UF. Patricio has been professor, what, 1 year?
Almost two
Almost
years.
2 years.
2 years.
But he has had an incredibly successful program, not only in terms of the grants that he's had, but also the students that he's been able to attract to this field. So, good to have you.
Thank you, Matthias. So thank you guys for staying for my talk. So there's been a lot of discussion and improvements in terms of genomic selection in diploids, but guess what? Autotetraploids or polyploids in general are being excluded from this discussion. So today that's the topic of my talk. Is going to be toward genomic selection, how we're dealing actually to implement genomic selection in autotetraploid species. But you're already walking among tetraploids or polyploids in general actually. So when you have tea this morning, or when you have coffee, or the sugar that you use in your tea or coffee, or the peanut butter maybe that you put in your bread, actually, those are products of a polyploid species. So it's something that they're already among you guys, but they are being excluded of this main discussion. But what are polyploids? So we have two kinds of polyploids. The autopolyploids, actually, the ones are generated by just reduplications of genome within a given species. And we have the allotetraploids, actually, there is an event of hybridization between two species and then the duplication of the genome. So today we're going to be concentrated actually in the autotetraploids. And just to give you an example of how behind are these species in terms of a study, compared with diploids in terms, for example, quantity genetics and population genetics. All of you probably know the book Falconer, Introduction to Quantity Genetics. It was published originally in the 60s. Well, the equivalent book for autotetraploids actually was just published in 2003. So that's to give you an idea, more or less, how behind are these species. And now that genomic selection is being implemented in diploid species, actually these species, the autotetraploids, are going to be left even behind. So that's what we're trying to correct. And that's what is the rationale, actually, that brought us together with Jill Olmsted from the University of Florida in charge of the blueberry breeding program, Jeff Endelman from the University of Wisconsin now working in the breeding program of potato, and myself working with alfalfa, the three of them autotetraploid species, or autotetraploid species, I say correctly, with the goal of actually accelerating the autotetraploid breeding by developing the capacity to implement genomic selection or using molecular markers to predict the traits that we are selecting for. And even when these species are so different, I mean, we are talking about a fruit that you consume directly, a potato that you process and you eat the products, and an alfalfa actually that you feed the cows, or eventually you eat the sprouts also in your salad. But all these species actually has been demonstrated that the rate of genetic improvement has been relatively low compared with diploid species. So, but how these species have a special, challenges. One of them is, for example, is the dosage effects that we'll explain in a second. The double reduction effect also. We don't know how those effects are going to impact genomic selection because they've never been studied. There's a problem that we have by working with these species, that is a lack of tools to do this kind of work. And in a separate point also, and only in the case of forages of alfalfa, we don't select individuals. We don't select inbred lines. We don't select hybrids, but actually what we select populations. So that's an additional challenge that we cannot use the traditional methodologies or tools that you are using in genomic selection nowadays for diploids, but we need to adapt our own tools to use it in autotetraploids. But before we go there, I just need to explain, and most of you probably know about this, but I just need to remember what I'm talking when I'm mentioning dosage. For example, in this case, we have different dosages from 0 to 4 doses actually of the allele B in this case. And we are talking here about autotetraploid species. The equivalent, obviously, is in the diploids, where you only have 3 possible states. So the monogenic for the A allele, monogenic for the B allele, the heterozygous. So in this case, I want you to imagine if you are doing a next generation sequencing project, and actually you see the 2 alleles, you will call it heterozygous. Well, in the case of autotetraploids, these 3 ones here are the heterozygous. So how do you differentiate from the simplex from the triplex? And what is going to be the effect of that in the prediction of the phenotype in genomic selection? On the other hand, we have double reduction in autotetraploid species. It's very specific to autotetraploid species, or autopolyploid species in reality. So what is double reduction actually is when sister chromatids from the same chromosome migrate into the same gamete. Basically, you end up with 2 alleles that coming from the same allele in a gamete. So even when there is recombination there, because recombination needs to exist actually for this to happen, but actually there is a double reduction in the process of meiosis, and basically you end up with a given allele getting duplicated but migrated in the same gamete. So the consequences of this is actually that creates inbreeding even when you are crossing 2 individuals that might be totally unrelated from each other. So what is the effect of this, at the same time, in the methodologies that we are using We don't know because it hasn't been studied. And that's what we're working with. So the first section of the talk is going to be about the dosage and how we are dealing with that. So this is a collaborative project, and in this section of the project, Jeff Endelman is working with his student Carly Smith, if I'm pronouncing that correctly. And the goal basically is to accurately predict or call the tetraploid genotypes SNP by using arrays and at the same time sequence-based methodologies, next-generation sequencing. In the first stage, where Jeff and his group, actually they are working with the Illumina array and using the intensity of the allele of the chip as a way to do this. Basically, this is already known. You can use the intensity of the two alleles actually to estimate more or less the angle that's generated by the intensity clustering that you see. And you can use this theta actually to also, you can put that in a graph like just plotting the theta and then in that way doing the calling of the genotypes. And you can do this manually. But imagine if you are doing a next generation an array actually with 4,000 markers or 6,000 markers, it will take you quite a while. I mean, the student doing this is not going to be very happy doing that work. But on the other hand, I mean, there is current other methodologies that you can use, for example, fit tetra or tetraploid map. And both of these methodologies actually use the same kind of algorithm, which is a mixture of models of normal distribution actually to do the genotypic calling. So as you see here, so it's a mixture of normal distributions, and in that way they do the calling. Now the problem with these methodologies is actually they have issues when actually one of these cluster is missing, or when you actually have double reduction. So in this example, we have two biparental families, potato families, and we have here the red family and the blue family, for example, and here we have the expected progeny of that cross plus what you will expect, for example, you have double reduction. And for the blue family, the same thing here. So here we have, for example, in this case, in the red family, the call by this methodology is good, and there is no problem with the double reduction. But in some cases, when it's a low number of individuals in a given cluster, a product probably of double reduction, you're going to have problems in the calling. Okay, so as Jeff pointed here, the mixed models have difficulty identifying small clusterings in the current methodologies. So his group basically developed a new methodology that's basically they call cluster call, that's based on hierarchical clustering, and what is able to deal better. I mean, as you see, everything is going to be similar, but it's able to deal better when for cluster actually that have low number of individuals in the cluster. So basically the accuracy of the calling is going to be higher. And actually that's represented here. Well, and this I probably need a second to explain you what we are looking at here, but we have the quality threshold in this axis, and here we have the accuracy of the calling. And the accuracy calling, in this case, it was three biparental families, genotype with 2,000 individuals each. And then the methodology was used independently in each of the family. And then they were looking across of the three family to see if there were consistency across the calling for the different genotypes. Okay? And basically, if they were accurate, they were score up. And then there were around 455 individuals. So there was a big jump actually in what you can, to a given threshold of accuracy, for example, there is a big jump in the number of quality callings for using the methodology developed by Jeff and his group. So that's great. Now, what is next in this area, in this section that we are working with, because we need to deal also with next generation sequencing data. So we have solved that, or Jeff group has solved that for the array. So we have 90% of accuracy in the calling. Now, how do you deal the next generation sequencing data. So in this case, something that we are thinking is actually you can use the number of reads as you have the intensity in the chip arrays, you can use the number of reads actually in the next generation sequencing as your intensity signal in order to do kind of similar things. So the second problem that we're going to be addressing also is that in this kind of methodologies that Jeffrey's working basically is using the 3 biparental populations to somehow train the system to do a better calling of the genotypes of future individuals. But what happens actually if you don't have those few biparental populations in order to train the system? So for example, in breeding populations or GWAS type of populations directly. Is the methodology is gonna be better, and that's something that's coming in that area. So the second part of the talk is going to be about it's very traditional in genomic selection that actually we have a baselines to compare our genomic selection methodologies, and usually this is the phenotypic blab. So guess what? In autotetraploid species, phenotypic blab is not applied. And it's mainly actually because of the existence of the ways to calculate the additive relationship matrix, so that you see here. So this is a traditional mixed model that we use with fixed effects, random effects, and the error. And the assumption that we usually do is that the genetic effects is normally distributed with a mean of 0 and a variance of sigma squared actually being multiplied by a relationship matrix, which is derived from the pedigree of the population in a certain way. That in animal breeding, this is being proposed, I don't know, in the 60s or even before. And in forestry, actually, it's been used since the 90s. In other diploid species, crop species, being used most recently. But in autotetraploids, actually, it's completely missing. Now, just to tell you, so how to calculate this matrix actually was just published in 2012. So what is the impact actually now on of the double reduction in this matrix, because these matrices are derived from the pedigree that you have in your population. But we know that the double reduction causes inbreeding. And just to give you a very quick and easy example here, so we have, if we have this pedigree scheme that you can also write kind of in a 3-way column or 3-column that we usually do for some of the programs that we use in quantity genetics. And in the traditional diploid matrix, for example, you will see the relationship between, for example, 2 and 3, because it's one of the partners, is going to be 0.5. And actually, if you can build the relationship matrix nowadays, we can build the relationship matrix for tetraploid species or autotetraploid species. And we also can plug different levels of double reduction there. So we can study what is going to be the effect of the double reduction in the prediction of the phenotypes. And in this case, for example, the same relationship between, for example, 4 and 3, there was 0.5. In this case now, it's going to increase by 0.55 if you have a 10% of double reduction in this case. So the double reduction increases the inbreeding and obviously the relatedness that you have in the population. So it's going to be a problem for breeding. But we have an additional problem for the people working in this area. There is no tools to create these matrices. So then we decide, okay, so we first need to create these tools in order to evaluate what is going to be the effect of double reduction in this population. So, and that's what it did. So this was led by, in my lab, and by an undergraduate student, actually Rodrigo Amadeo, who is now back in Brazil. He worked a little over a year in my lab as an intern. And basically we create a package, an R package, that you can build the relationship matrix for autotetraploid species using the pedigree, but at the same time you can build the relationship matrix out of pedigree, genomic data, or combination of them for diploid species. And we are expecting also to include the autotetraploid genomic relationship matrix, so we can use the GBLAB methodologies to observe what is the effect of the double reduction in our predictions. And that package probably is going to be released in this coming fall. Now that we can build basically the relationship matrix with different double reductions, now we need to set our baseline for the first time in this species, and in this case, the first species that we're working with is the blueberry. Like I told you before, is the program of Jim Olmsted here in the University of Florida. Blueberries are, very important fruit, and actually the consumption has been scaled tremendously lately. And one of the reasons actually is because it's considered a very healthy fruit. So a lot of people is trying to consume blueberries, and it has a lot of benefits. And Jim always showed me, the number of citations and new papers actually that shows the new benefits of blueberry, and then the list is already very large, even to include in the paper. So the UF cultivars, the University of Florida cultivars actually have been growing in every continent except in Antarctica. And actually 95% of the acreage here in Florida. Since 1976, for example, 39 cultivars have been released from this program in the University of Florida. But most impactfully actually is other numbers that Jim showed me, actually that 50% of the cultivars released in the last 5 years actually have been directly by the University of Florida with germplasm that came from the University of Florida. Of all blueberry cultivars actually, not only the ones from UF. So this program actually used phenotypic recurrent selection since the 50s to the present. And as you might know, recurrent phenotypic selection, it might take very long. So these are methodologies that actually take a long time. In this case, up to 15 years to develop a new cultivar. So obviously the benefit of genomic selection in these species that are perennial, and with a long cycle of to generate a given cultivar is going to be tremendous. And we are envisioning in this case, after going after several meetings and defining where does actually genomic selection will fit better in this kind of population, that actually will be around between year 3 and 5 in these cases. So to start with, so in that case, actually, between year 3 and 5, actually, in any year, Jim is going to be dealing actually with 2,000 individuals in his breeding population. And they will be derived more or less from 100-something crosses, in this case 124 crosses, from more or less 100 to 150 different parents. So in this case, this is the training population that we're using now, just to set the baselines, and later we're going to be doing also genomic selection. So the important thing is the training population in this case represent the whole germplasm of the breeding program. Okay? So we are not creating a new training population to try to do this. There's already been many cycles of selection, so relationship in these populations is already there, so the linkage is already there. So we want to study what is the effect of double reduction in blueberry in the genetic parameters. And this is the work actually that's been done by Catherine Ceylon, which is a student under supervision of Jim and I. And for this we're using the training population I was showing you before, and we're using a very extensive pedigree. So they have 7,755 lines in this pedigree, and this pedigree data dates since the 1908, actually. And that was one of the part of the works of Catherine to basically reconstruct, with the help of Jim and others, reconstruct the whole pedigree basically back to the founders. So with this pedigree, actually, we build different relationship matrix with different levels of double reduction. We feed all these models later with the same phenotype, so the phenotypic data is the same that actually we collect from the field, and we're using different traits from yield, quality, and others. And we feed these models actually in HREM in a traditional phenotypic BLAP scenario. The only thing that changes between these models actually is the level of double reduction that's included in the A matrix. Obviously, we calculate the AC, or just a log likelihood of the data to fit the model, or the model to fit the data. And we compare with the diploid model in this case. And these are the results for a few of the traits that we are working with. So as you see, and this is actually here, this is a kike information criteria of the IEC, the difference from the diploid model. So the higher is actually the better in this case, because it's the different from the diploid model. Okay, so as you see in this case, for most of the traits, actually, the level of double reduction that's being estimated, or that actually that fits better the data, is 0. We still need to use an autotetraploid relationship matrix, but the level of double reduction there is going to be 0. That's the one that fits better the data. But actually for one of the traits, that's firmness in this case. This is the firmness of the foot. And in which ones the level of double reduction that fits better the data is actually between 0.15 and 0.20. Now we know that the maximum theoretical level of double reduction is around 0.16 to 0.17. So actually that's the number that we are calling in this case. But what is the effect of double reduction, for example, in the genetic parameters? And that's what I'm showing you here. So this is the estimation of the heritability by increasing the level of double reduction. And as I told you before, and this is for most of the traits, even when this one is for foot firmness in this case, for most of the traits, there is not going to be a big difference between fitting actually a traditional diploid model that's, is wrong in the sense that it's not the right model for this species, but fits very well. But by fitting a tetraploid relationship matrix in the same model, it decreases a little bit the heritability, but it's not significant. And this also has been observed in other studies in potato, actually. But actually, as you increase the level of double reduction, and more or less for most of the traits, the trends was very similar, there is going to be a decrease or a loss of heritability due to this increase in the inbreeding in the relationship matrix. Most importantly, we ask as breeders what happens actually if you have different levels or different depths of the pedigree that you are using to reconstruct this, or to find out what is the best double reduction that you have or what is the real double reduction that you have in your population using this kind of methodologies. Because that could be a problem. In some pedigrees, actually, you could reconstruct the whole, in some programs you can reconstruct the whole pedigree, but in others you will never be able to do that. So what happens if I start doing this just with a shallow pedigree that I reconstruct a couple of generations before? So as you see, this is the figure I just showed you before of the fitting of the data given different levels of double reduction for different traits. And actually, as we start shrinking or putting thresholds in the pedigree of the amount of pedigree that we're using, we start seeing a change in the pattern actually of what is the double reduction that you have in your population. The most extreme actually being that from no double reduction when you use all the pedigree and the real data that you have in your program, and actually if you use a very trunk amount of pedigree in your estimations, the level of double reduction that you estimate actually to be maximum, about 0.16 or 0.17, which is the theoretical maximum. So the take-home message is here is that we are creating this tool. It's going to be released for everybody to use, but you need to be using carefully. I mean, if you don't have a complete pedigree, actually you might get levels of estimate the level of double reduction in your population that might be wrong. So in this area, we're also going to be testing these methodologies in the alfalfa and potato populations, because remember this is a collaborative project with 3 species. We're going to obtain the genomic information for the blueberry population. Now this is very key and is important. We are working with very complex species here. And remember, some estimations that Jeff did actually for the project show that we need around between 40 and 60x coverage or depth in the sequencing in order to actually differentiate the duplex from the triplex or the simplex bit and the triplex. So, and at the same time, these species, they don't have a lot of genetic resources, so there is not much information. So in this case, we are using sequence capture to sequence these species, and we just got the first 96 individuals genotype with around 30,000 probes, and we obtained around 166,000 markers, which is by far, I mean, probably the most markers that you have in this species in the order of a few thousand now. So it's by far the larger number of markers that have been sequencing, have been produced for this species, and then we're going to be doing the auto-deploy genomic matrix to basically evaluate what is going to be the performance of the genotypic blood now in this species. Now, the last thing that actually is, as I was telling you before, in most species, I mean, in other species, not in most species, in some species actually the selection target is not going to be an individual, it's not going to be a hybrid, or it's not going to be a given inbred line, but actually it's going to be a group of individuals, a population, or in the case of forage, a synthetic. So in this case, by using the methodologies that we were just talking before, it's not going to work, because basically in this little plot that we have here, we have dozens of different genotypes. Okay, or the same in this case. So this is the case of alfalfa or ryegrass, and actually this is a diploid species, so it's not a problem actually of autotetraploid species. It's also a problem of some of the diploid species. So the target, the selection target, is not going to be the genotype, and on the other hand, even when the cost has decreased significantly for next generation sequencing, you know, it's still too high for public breeding programs, for example, the ones I lead in now. So I cannot do the methodologies. I cannot sequence, you know, dozens or thousands of individuals in order to do the traditional genomic selection that we have been working with for so long. So in some cases, it's justified because of the value actually of the target individual that you are selecting. But in others, the value of the target individual, or in this case of grouping individuals, it will not justify application of genomic selection in the traditional way. So this is a part of the work that is under Esteban Rios, a PhD student working in my lab, and also Mehul Bhakta, a postdoc working with the support of Mehul actually, a postdoc working in my lab. And the idea here is that because we don't have the data yet for this book, genotypes of all populations, but we are using the pine breeding population that we've been working extensively in our group. And in this case, we have 71 different families, and this was genotyped with a chip of more or less 5,000 or 4,700 markers. And the idea of this methodology, actually, that we're trying to propose here is basically use the phenotype. The phenotype is going to be an average of the individuals in a given family. So, and the genotype is going to be the allele frequency actually in a given family. So, just to give you an example, so you're going to have now, you make a cross, you have a cross of individuals, and you might have hundreds of individuals, for example, in the species that we work from a given cross. And then what you're going to do in that one, you're going to select several leaves from each of the plants. You're going to get the DNA, you're going to get the molecular markers, and then you're going to sequence that, and let's say in next generation sequencing, and then you can estimate the allele frequency for that given cross. So you are not doing it for every single individual in that cross. So I told you you have 100, so imagine genotyping 100 individuals, and actually we are targeting only one cross. But in this case, actually you're going to be doing only a sequence of a single bulk or a single individual, if you want to call it like that. And now in the case of the phenotype, in some species that we are working with, we already take the phenotype as a bulk. We already take the phenotype of the plot. But actually in this case, what we are doing is actually we are doing this phenotype in silico. All this work was done in silico. Basically, we are bulking the phenotype and we are bulking the genotype in order to make this work. And I'm just going to jump to the results right away. So basically, it's interesting actually by doing it in these ways, and even when you're doing we're using 71 families only, the prediction ability that you have is larger than what you have in the individuals. Now, but there is a catch. It's too good to be true, no? So, and even the important thing here is that actually this methodology is capitalizing the information at the mean level. And the information at the mean level always going to be more accurate than actually the information at the individual level. And that's known. In the quantitative genetics, we know when we do, for example, parent selection or when we do family selection, we know that's more accurate actually than doing individual selection in the animal model or in the individual model. Now, the catch I was telling you before that with the individual model, actually, you are targeting 100% of the genetic variance. Well, in this methodology, you are only targeting 50% of the genetic variance, because remember that we're just studying the difference between the different families here, which is going to be 50% of the genetic additive variance. And in the case of the individual, you are trying to target 100% of the genetic additive variance. Now, to make it comparable, then we can just multiply by the square root of 0.5, which is the parentage between full SIF individuals. And actually, even in that case, we're going to obtain that actually in this set of traits, we're still going to get that our methodology is going to be, in 6 out of the 8 traits, is going to be better, actually, the traditional genomic selection. So we also did this in eucalyptus and other species with different traits and different architecture, and in this case 68 different families and some hybrids also in this population. And in this case it was sequenced by sequence captured by rapid genomics, obtaining 500,000 SNP markers. But in this case we use a subset of that, around 50,000, 20 to 50,000. And we also observe the same thing, and in some cases actually gets very close to the individual selection. And we might think that actually there might be problems of the phenotyping in those cases of the trait, because that's going to have a high impact in the accuracy. But we did the same thing here. Also, we multiply by the corrector factor to be comparable. And in this case, out of the 13 traits, when you compare at the individual selection level, it's going to be, this methodology is going to be better. So while doing this, Esteban was doing some runs one day, and we're trying to do as many folds in the cross-validation, because all this was done in cross-validation. And in some of the runs, we will observe actually that the accuracy will be good, the good average, good average, good average, and in suddenly it will drop to 0 or negative values. So I have seen several studies actually where that has happened, and they have gone to see, okay, is actually the relationship? And John showed a little bit of that before, just how the increase in relationship between the training and the validation increased the accuracy that you're going to obtain. And actually we went there, we studied, the subset of each of these training false subsets for the relationship with the training population, and actually we couldn't find anything related to that. So that was not driving the accuracy in this case. And we also did, Esteban also did some clustering, some clustering of the methodologies, and we find some more or less 4 groups by doing the clustering on the phenotypes, and by fitting those different groups, we couldn't answer why also it was lower. He also studied the allele frequency, and more or less the allele frequency in some cases it gave us partial answer, because in some cases actually we find out we have families with very low number of individuals, and those ones were giving us problems, because actually the accuracy for those given families was very bad. Now, if you are doing this at the population, at the family level, so you want to estimate the phenotype and the genotype as accurate as you can. So how are you going to estimate the phenotype as accurate as you can if you have many individuals in that family? And the same thing with the genotype. So if the less individuals you have, the higher the sampling error that you're going to have for the given genotype of the higher sampling error in the allele frequency for that given family. So after doing several work, actually we realized that we need to drop all the families with less than 5 individuals, and that gave us some good results there. And I'm not going to show that. But, and actually those are the results that you saw before. They're already been filtered by families with less than 5 individuals. But what's interesting actually that in one point we start looking at the distribution of a given trait. For example, I think this was DBH. And then we divide the population in 3 sections. So the bottom 10%, the top 10%, and this trait has been standardized, of course. That's why you see that scale there in the x-axis. And then we have 80% of the population actually in the middle. So what we did is actually we did some training and validation using these different portions, as you see here in the bottom, in the two different in the sea clone pine species and the eucalyptus species when you use the bottom, and when we use the top, or when you use the middle. And actually, when we say combine, here is a combination of top, bottom, and middle, and only we have errors in this case because you can do it multiple times. If you select the 10%, you only have 71 families. You are selecting 7 families, and that's it. And also for the top 10, so you don't have repetition in that case. But actually, for the combine and the middle, you can do multiple replicates. And I remember I have seen some of these also in the past. Actually, I think it was Guillermo Rosa that showed us, basically, if you don't have a good sampling of the allele population that you have in your population, actually, you're not going to be able to have a good accuracy in the prediction, and that's what we were finding. So in this area, actually, we are doing some simulation now, and we have the simulation. We just need to partition the population to study, because it's kind of showing us some different patterns here. So we need to study, the number of families that we need to include, the number of individuals in a given family, what is going to be the effect of the heritability of the genetic architecture as well in this type of evaluation. And at the same time, we need to evaluate the bulk genotyping, because what we did before, it was just in silico, so basically we did everything in the computer. But we're already working on that, and my whole BACTA, the postdoc I was telling you before, is already doing some bulk genotyping with very good results in this case. So just to summarize, in the progress toward genomic selection in autotetraploid species, we can tell you that we have made some progress in the last year. The dosage calling is a big improvement in the arrays, and now we're going to go to next generation sequencing, no, Jeff? And we also find the ways to establish the baselines in order so we can compare what's going to be the effect of the genotypic blood, for example, or the genomic selection in these methodologies. We're being able to estimate the effect of double reduction in at least in the blueberry population. We are developing different tools, for example, the cluster count and the AGH matrix that are going to be released in this coming fall. And we also, this is more related to some species only, but we also know more or less what is the effect of doing both family bulk genotyping and using that information, genotyping and phenotyping, and using that information for genomic selection. Most likely a methodology that we're going to be using in my breeding program, because as I was telling you before, if I have 100 families and each of them you have 100 individuals, it's already impossible for us to do the genotyping. Now, if we have those same 100 families, I just need to genotype 100 samples with this new methodology and find out what are the best families in those cases. And remember that the other 50%, once I identify the best family, I know that the other 50% of the genomic additive variance is enclosed within that family. So I can just go and explore that phenotypically, for example, or with another methodology. So this is something that's already still in progress of development. So with that, I would like to acknowledge the collaborators and the graduates, undergrad students, and postdocs that are being working in this project, and obviously the funding agency in this case, because without them, we'll not be doing this work. Thank you, guys. Yes, but remember I was telling you before that not much genomic data existed in this species before. I mean, it's something that we're just developing, and Jim actually in the blueberry, he was doing some. He did some GBS actually, but the results are still not very reliable, and now with the sequence capture data, we expect to do that in that population. But I know also Jeff did some genotyping in the potatoes, and is still working on that, if I remember well. And we are genotyping actually this fall. So, but yes, that's kind of the ideal, that you do it with the genomic data. But in order to do that, actually, you need to have very reliable data. Because if you think, for example, if you have an error, you might, I mean, if you're looking for actually for an allele change in the nucleotides. So let's say from one error in the genotypic platform is going to give you a wrong answer, for example, in the double reduction that you might have. Now, the cumulative, maybe you're going to be able to clean that, but still you need the most reliable the genotypic platform, the genotyping platform you use, the more reliable you're going to be able to get what is the real level of double reduction in your population. That's what we're expecting to do now.
Curious about the Tetrapoid. Are we going to need to have much larger training populations in order to get the best of the
That's a good question, and that's something that actually that we also going to be investigating, that we don't know yet. I mean, in the traditional genomic selection, if you think for diploid species, the larger the population size, the better is going to be the accuracy. Now, in this case, in the case of the autotetraploids, the same thing is going to apply. But now, should it be larger than diploids? You have, I mean, the theory will suggest that yes, because you have more states on a given genotype, no? But it's something in reality that we haven't done yet, so we cannot answer.
Why do you think this accounting for double reduction doesn't increase your model?
Accounting for double reduction doesn't.
So some of your results will be old heads.
Yeah. The best one, yeah.
Then it said you put 0 double reduction.
Well, because basically it's telling us that there is no double reduction in this species. So that's what it's telling us. It's not that it's not that we are accounting or not by double reduction. The way that we are doing this is trying to, by seeing how the data gets fit by different levels of double reduction, try to identify what is the real level of double reduction. And in that case, basically, the model is telling us there is no double reduction for most of the trait in this species. And we have been discussing with Jim what are the implications, I mean, why this is happening. And one of the requirements to have double reduction is actually to have multivalent information and in meiosis. And the chromosomes arms, they need to be long enough actually for that to happen. And in the case of blueberry, that's not, that doesn't happen. Now, maybe in the case of one of the traits, in that case, if that's not the explanation, in the case of because you have one trait actually is showing double reduction, maybe then the alleles controlling the other traits where you find no double reduction might be close to the centrometer in the chromosomes, because double reduction is also position dependent in the chromosome. The closer you are to the centrometer, the less double reduction you're going to have, because the less chance that you have recombination and the two gametes migrate, I mean the two alleles migrate to the same gamete. So.
Questions? All right. Thank you, Patricia.
Thanks.